Welcome, and thank you for standing by. At this time, all lines are in a listen-only mode until the question-and-answer session. At that time, if you'd like to ask a question, you may do so by pressing star, then 1, and recording your first and last name. Today's call is being recorded. If you have any objections, you may disconnect at this time. I would now like to introduce your host for today's call, Ms. Irene Ihier. You may begin.
Hello, and welcome to today's FDA webinar. I am Irene Ihier of CDRH's Office of Communication and Education. As part of the FDA's ongoing effort to assure patients and providers have timely and continued access to safe, effective, and high-quality medical devices, we are hosting today's webinar to provide investigators, sponsors, and new developers with information on the regulatory landscape for neurological devices. This webinar is part of the FDA's partnership in the White House Brain Initiative, focused on understanding the human brain and uncovering new ways to treat, prevent, and cure brain disorders. Dr. Carlos Pena, Director of the Division of Neurological and Physical Medicine Devices in the Office of Device Evaluation here in CDRH, will begin today's presentation. He is joined by members of the division. Following the presentation, we will open the line for your questions related to the information presented in today's webinar. Additionally, other center subject matter experts will join the team to assist with the Q&A portion of our webinar. Now, I give you Carlos.
Thank you, Irene. Good morning to all those on the phone. My name is Carlos Pena, and I'm the Director for the Division of Neurological and Physical Medicine Devices. And I'd like to welcome you to our first FDA regulatory webinar for investigators and sponsors of neurological devices. This is a 90-minute session comprised of a couple parts, including a brief primer on medical device review and regulation, non-clinical testing, early feasibility studies, and best practices on engaging with FDA. Our goal is to brief all attendees on each of these points, but most important, sponsors and investigators obtain a sense of how to navigate the regulatory landscape of medical devices so that our landscape is more transparent, predictable, and efficient, and thereby assures patients and providers have timely and continued access to safe, effective, and high-quality medical devices. This webinar also supports, as Irene mentioned, our support of the White House Brain Initiative, which is focused on understanding the human brain. I'd also like to mention that this webinar is hosted not only by myself, but several talented staff in the Division of Neurological and Physical Medicine Devices, one of the newest divisions at the Center for Devices and Radiological Health at the FDA. So at the FDA, the vision of the Center for Devices and Radiological Health is that patients in the United States have access to high-quality, safe, and effective medical devices of public health importance first in the world. We take this principle seriously with urgency, and we hope to show you how serious we are with some hard data at FDA. A medical device, for some background, is defined as an instrument, apparatus, implement, machine, or related article intended for use in the diagnosis or cure, mitigation, treatment, or prevention of disease, or intended to affect the structure or any function of the human body and does not achieve any of its primary intended purposes through chemical action. And so you might be saying, Carlos, there are several provisos, addendums, clarifications. What really is a medical device? And in short, if it diagnoses, treats, or prevents disease, it's a medical device. It also does not achieve this purpose through chemical action, and one can classify a device as a medical device even in the absence of claims when a device impacts the structure or function of the human body. We have been engaged in this technology sector some time, and this is a favorite slide of mine. Here I show you an array of products with neurological indications, beginning with neurothrombectomy devices, to epilepsy, DBS, neurodiagnostics, prosthetics, to therapeutic devices for migraine and microcatheters for the neurovasculature during surgery. Many of these products treat a disease or condition. The goal is not to discuss individual data in support of each device, but share with you here that each device went through a regulatory path that was in part tailored to the individual risk and benefit profile of the device. And when other products may be targeted for the same indication or used in combination, we regularly, often, and without hesitation, engage our colleagues in other product centers. Medical devices are classified into Class 1, 2, and 3, with regulatory control increasing from Class 1 to Class 3. The device classification regulation defines the regulatory requirements for a general device type. And so, for example, most Class 1 devices are exempt from submitting an application to FDA, most Class 2 devices require pre-market notification, and most Class 3 devices require pre-market approval. We provide oversight across the three classes using tools known as general and special controls, for which we help to communicate to sponsors and investigators to help them meet their regulatory obligations. As mentioned in the last slide, medical devices can be classified into three types, two of which are listed here, Class 2 and Class 3. And these higher risk classifications can be linked to regulatory submission pathways. For example, we receive several dozen PMAs each year. These submissions are the highest risk and typically require clinical data. These are Class 3. The second pathway is the 510K submission pathway, and we receive several thousand each year. They typically do not contain clinical data, but are supported by non-clinical data and bench testing and review of prior submissions that may have contained clinical data. These are typically Class 2. And finally, a third regulatory pathway is the de novo submission process, which includes devices that aren't comparable to anything on the market and presents a low to moderate risk other than other types of devices. Typically, once we have granted a de novo, that particular product becomes the predicate to subsequent products, which can then move along the 510K pathway. And de novo submissions will be the subject of our next webinar early next year. Another question that might arise is when is clinical data needed, especially when human studies need to be initiated and are significant advanced investments by sponsors and investigators, which FDA realizes. Typically for PMAs, clinical data is needed. For de novo submissions, typically clinical data may be needed, but may not always be needed. And for 510K submissions, clinical data is typically not needed, although there are cases where clinical data was submitted. So you're probably saying to yourself again, Carlos, there are a couple of provisos, addendums, and clarifiers. How do we find out what is needed? And my response is that you can request feedback on any of these questions through the QSUB process, through pre-submission, preferably before starting the study or making any investment into your device technology development plan. And we will return to this shortly when Tim Marginen reviews best practices on how to engage the FDA. One very important step in making sure sponsors and investigators understand the options that are available to them to get their product to market and making very clear and transparent the regulatory landscape. Here, several guidance documents that are relevant to neurological devices listed include pre-submission guidance documents that allows for an opportunity for early discussion and feedback from FDA, to early feasibility studies, design considerations for pivotal clinical investigators, and expedited access regulatory pathways for devices. And I'd like to note that the approach to early feasibility studies will be discussed also during this webinar by Erin Keegan and Debjani Saha. All the guidances are intended to clarify as clearly as possible the considerations that should be addressed to hopefully move a product to market and ultimately to patients. And FDA is considering additional guidance, and I'm delighted to indicate earlier this year we released a draft guidance for public comment, not for implementation, focused on conducting clinical trials on medical devices targeting neurological disease progression and clinically meaningful patient outcomes, a novel guidance that aims for regulation to keep pace with innovation. Many paths to market include collecting clinical data about a medical device, and they usually occur under what's called an IDE, or an investigational device exemption, comparable to the drug side of an IND. IDEs allow an investigational device to be used in a clinical study in order to collect safety and effectiveness data required to support a marketing submission. And FDA has 30 days to make a decision about any given IDE, and we strive to get an IDE up and running because the faster you can get an IDE going safely in the United States, the faster you can collect the data you need to submit a marketing application to get a device to U.S. patients. And before you get an IDE, we may ask for non-clinical data, and Molly Ghosh and Danya Williams will be discussing with you our non-clinical requirements and recommendations next. But first, I would like to share with you how serious we are about moving devices to patients. Here is a graph that shows decreasing timelines in getting a medical device study up and running. In FY11, the median time it took to get full approval took approximately 400 days. Fiscal years 2013, 2014, 2015, where we have reduced this time by more than half each year with goals, and the most recent goal being met in 2015. And I'd like to stress these are appropriate decisions with patient safety of paramount importance. One key change has been the interaction we engage with sponsors and investigators. I will also say that this has not been an easy transition, and there has been a cost to staff engagement in this effort. And we continue to look for ways to make this progress sustainable long term. This webinar aims to help sustain these numbers by helping to educate sponsors and investigators and developers and innovators so that they are able to start up their studies as quickly as possible, understanding FDA's expectations to getting those studies up and running, and we can approve these studies safely in the U.S. Take-home message is that we want to accelerate getting devices to patients who desperately need them, and starting those studies in the U.S. is an important critical first step. Last, I'd like to mention that our Division of Neurological and Physical Medicine Devices is one of the newest divisions in the Office of Device Evaluation at the Center for Devices and Radiological Health. The office is the primary point of the device evaluation for medical device sponsors and investigators to engage the agency, and we receive submissions for investigational studies or market approval in the U.S. on the right side called ODE, of which there are 7 divisions within this office, which is the last row, and for which neuro and physical medicine is one. Our division is growing, and next month we are moving from 3 branches to 5, including the neurostimulation devices, neurology and psychiatry branches, the neurointerventional and neurosurgical devices branches, and the physical medicine and rehabilitative devices branch. And you will find no better team of individuals positioned to safely move devices to the market fastest. I'm a little biased. These 5 branches take on the lion's share of products dealing with neurological devices. And with that, I would now like to turn to the non-clinical testing segment of this webinar with Molly and Danya.
Thank you, Carlos. Good morning, everyone. My name is Molly Ghosh, and I'm a toxicologist and lead reviewer in the Division of Neurological and Physical Medicine Devices at CDRH. Today, I'm going to talk about the biocompatibility assessment for neurological devices. Here is my presentation outline. First, I will provide an overview of biocompatibility of assessment of medical devices in general, and then I will discuss some special considerations for neurological devices, and finally, I will provide an example of biocompatibility assessment for brain implants. So there are 2 important considerations for non-clinical review of medical devices. One is performance. The question is the device going to perform as intended? The other one is about safety. Is the final product going to be biocompatible? For today's talk, I will focus on the biocompatibility assessment. So what is biocompatibility? Biocompatibility of a medical device refers to the ability of the device to elicit the desired biological response without causing harm in the body. So it depends on the body's response to the device as well as device response to the physiological environment inside the human body. So the next question is, why do we ask for biocompatibility assessment? We don't always know everything in a final product. Also, it is important to know that CDRH regulates medical devices in its final finished form. It does not regulate the individual materials that are used to fabricate the device. As you all know, that biocompatibility assessment is a critical part of medical device safety evaluation, and we recommend the assessment to be done on the final sterilized if applicable device. And the reason is, there could be different sources of toxicity from a medical device. It could be leachable chemicals or manufacturing residuals from the device. It could be potential degradation products, or it could be some interaction between the leachable chemicals. Also, the surface geometry, chemistry, and other properties can also adversely affect the biological response. So an assessment of potential biocompatibility risk from a device includes assessing the effects of leachable chemicals from the device, as well as the assessment of biological response to the surface properties and geometry of the device. Also, another consideration unique for the device is how biological response affects device performance. So ISO 10993 series of standards are widely followed for biological evaluation of medical devices. There are 20 parts to this standard. Some of the parts are very specific in testing guidance, while others are general. ISO 10993 Part 1 provides guidance on biocompatibility assessment strategy. It describes the device categorization and then recommends the assessment based on the category of the device. So I'd also like to mention here that in June of this year, we issued a guidance on the use of ISO 10993 Part 1, and this is actually going to be implemented today. So I will strongly encourage to review this standard if you are planning any submission to FDA. Also, I would like to mention about CDRH Consensus Standard Program, and I provided a link to the guidance document on the recognition and use of these consensus standards. So as I mentioned earlier, that ISO 10993 Part 1 describes device categorization. So for biocompatibility assessment, the devices are categorized based on nature of tissue contact and duration of tissue contact. So based on the nature of tissue contact, the devices are categorized as surface contacting device, external communicating device, and implant device. Based on the duration of tissue contact, the devices are categorized as limited contact, prolonged contact, and permanent contact. The limited contact devices are those that the contact is up to 24 hours. Anything beyond 24 hours up to 30 days is considered prolonged contacting device, and permanent contacting device has a contact duration longer than 30 days. The device categorization actually facilitates the selection of the appropriate biocompatibility endpoint for assessment. For example, the biocompatibility assessment requirement for a permanent implant like a deep brain stimulator would be a lot more than a surface contacting electrode, which will be used for probably less than 24 hours. So there is really no single endpoint that can predict the biocompatibility of a device. The biocompatibility risk from a device could be short-term, like some acute systemic toxicity or irritation, to some long-term effects, like chronic toxicity or genotoxicity. So here is a list of the biocompatibility endpoints that you may need to consider depending on the category of the device. And as I mentioned earlier, that the extent of assessment depends on the device category. Now, I would like to mention also here that some of the endpoints, like systemic toxicity endpoints or genotoxicity endpoints, can be assessed through a chemical characterization risk assessment approach on the final finished device. For some other endpoints, you may consider testing if the existing data could not be leveraged. So that's about some basic information of the biocompatibility of medical devices in general. Here, I will provide some special consideration you need to think of when you are assessing the biocompatibility of neurological devices. So here are some examples of neurological devices. This is no way a full list of diverse array of products that we see. These are just some examples. And as you see that there are some base materials which are frequently used for this kind of products are listed in parentheses after each of the product category. I would like to mention here that most of the time the biocompatibility issues that we see actually are not from the base material itself. It can come from the manufacturing residuals or some of the processing additives used during the manufacturing of that device. And that's the reason we recommend why the assessment should be conducted on the final finished device. For example, you all know acrylamide is a known neurotoxin, and this is used in some polymer production. So if there is any residual monomer in the device, it might cause some toxicity. This is just an example. So there are some special considerations for assessing the biocompatibility of neurological devices. As you all know, that the brain is protected by the blood-brain barrier, but when you are putting an implant in direct contact with neural tissue and cerebrospinal fluid, or CSF, it bypasses the protective function of the blood-brain barrier. So there is a need for animal implantation testing in relevant neural tissue to assess the neurotoxic effects. Also, we recommend that any devices in contact with neural tissue and CSF should be non-pyrogenic, meaning it should not cause any fever response. There are two sources of pyrogen. One source is the chemicals from the device that can cause fever response, and that can be tested by the rapid pyrogen method. Also, there is another source called bacterial endotoxin, which can also produce a fever response, and that is assessed through the sterility assessment using the LL testing. Also, it should be noted that any implant or any devices in direct contact with CSF and neural tissue should have a endotoxin limit of 2.15 endotoxin unit per device or less. As I mentioned earlier that, chemical characterization risk assessment approach could be used in lieu of testing for assessing some toxicity endpoints, but there are some limitations, and this approach may not be relevant for assessing local tissue response. Also, this chemical characterization risk assessment approach may only be used to assess the potential neurotoxicity of chemicals from neural implant if the toxicological data on the chemicals are available from the study where these chemicals were administered through the clinically relevant route of exposure. So here I'm going to provide an example on what are the factors or endpoints to be considered if you are assessing the biocompatibility of a permanent brain implant. So for the device category, it's considered a permanent implant with neural tissue and CSF and indirect contact with blood. So you need to assess the biocompatibility per ISO 10993 Part 1, and I have listed some of the endpoints that need to be assessed. In addition to these general biocompatibility endpoints, neuroimplantation study may also be considered to assess the neurotoxicity due to brain contact. Also, as I mentioned earlier, the endotoxin limit should not go over 2.15 endotoxin unit per device for this type of devices. So also, I would like to mention that currently there is really no standard protocol for a brain implantation study in the ISO 10993 standards. There is a brain implantation study protocol being developed, which will be included as an annex in ISO 10993 Part 6, which is the ISO standard for implantation. But currently, the standard which is available does not have this protocol yet. So here are some considerations if you are planning on doing a brain implantation study for neurotoxicity assessment. So if you are using a passive implant for your study, you might consider small animal models like rabbit models. Other consideration could be separate test and control group, equal number of male and female animals in the study, and the inclusion of different time points to assess both acute and chronic responses. Also, the study assessment parameters may include clinical observation, neurobehavioral assessment, and histopathology to look at neurodegeneration, gliosis, and myelopathy for the neurotoxicity assessment. You can also, you may also include some of the systemic toxicity assessment endpoints like body weight change, food consumption, clinical chemistry, hematological parameters. So this is just an example of if you are planning on doing a brain implant study, these are some of the points you may want to consider. So lastly, I would like to mention also that, you know, if designed accordingly, your an animal performance or functional study can be used to address some biocompatibility endpoints. Our next speaker, Tanya Williams, will be talking about the animal study, but here I'm just going to provide you a theoretical example. For example, you know, you are developing a new deep brain stimulated system and you are planning on doing a large animal study with the active implant to probably evaluate some of the stimulation parameters. What you can do also that you can actually design this study to assess some of the biocompatibility endpoints like implantation, some of the systemic toxicity endpoints. For example, you may consider including control groups like you know, sham surgical control groups to assess the effect of surgery on tissues because, you know, surgery causes tissue trauma. You may also consider including a control group with the passive implant to look at several, look at the material-mediated effects of the implant. Different time points may be included in your study design to look at acute and chronic responses. Also, clinical observation, behavioral assessment, and histopathology with specific stains to look at neurodegeneration, gliosis, myelopathy could be included as a part of the neurotoxicity assessment. Also, some of the systemic toxicity could be assessed through clinical chemistry, body weight change, hematology, etc. So that's, I would like to mention that if our animal studies designed properly to assess some of the endpoints, you may not need a separate biocompatibility study for implantation. So I will stop here and then hand it over to Tanya. Thank you for your time.
Molly. Hello, my name is Danya Williams, and I'm a biologist and a scientific reviewer in our division. This section of the webinar will focus on non-clinical animal studies. So first, what is the purpose of conducting an animal study? Animal studies are intended to demonstrate that the device under study is sufficiently safe for early human experience, for example, to support an IDE application or demonstrate device safety in support of a marketing application while incorporating modern animal care and use strategies. An animal study is warranted when there are performance evaluations or identified risks that cannot be addressed through bench or in vitro testing. For example, the effects of the technology or materials on brain function or neurotoxicity. Good laboratory practice, as outlined in GLP regulations 21 CFR Part 58, applies to non-clinical animal studies to assure the quality and integrity of the safety data. GLP regulations also outline the basic elements that should be included in an animal study report for regulatory submissions. Non-GLP studies may be acceptable in some submissions. However, sponsors should provide adequate justification as to why GLP provisions were not met how this study deviated from GLP, and why these deviations would not impact the study outcomes. For example, if an independent quality assurance individual is not utilized to periodically monitor various phases of the study, scientific justification should be provided to describe how the functions of the quality assurance unit were performed in order to ensure the validity of the data and that the study was conducted in accordance with the signed study protocol. Also, please note that it's important to provide a full study report with your submission with a complete analysis of your data for review. In this section of the webinar, we're going to be looking at some of the aspects that should be taken into consideration when designing a non-clinical animal study, such as animal model selection, study size and duration, and study endpoints. It is important to plan a study in accordance with the device's indications for use and any planned clinical studies. This can really act as a roadmap for designing your animal study to address identified risks and to use procedures to conduct the study under clinically simulated conditions when possible. When selecting an animal model, it's important to provide scientific justification for the animal model that's used. An animal model should generally be accepted for the study of the device type. This may be based on the utility of an animal model for a product class based on scientific evidence or literature. It is important to take into consideration the clinical relevance of an implant or treatment site and understand the possible local and downstream responses. Depending on the device, it may be recommended to use a simulated model to assess safety and or performance. For example, the use of a simulated aneurysm for aneurysm devices or a simulated clot for neurothrombectomy devices. There are some expected limitations to the use of animal models. For example, the tortuosity of neurovasculature of the animal model compared to humans, or maximum defect sizes or size limitations due to the physical characteristics of the animal. It is important to acknowledge or address these limitations. There's a great diversity in animal models that are used for neurological devices, ranging from rats and rabbits to sheep, swine, and canines. For example, in the past, swine have been used for neurothrombectomy devices, and canines and rabbits have been used for flow diverters. When designing an animal study, study size and duration are important aspects to consider. Studies should include the minimum number of animals necessary to generate valid and meaningful scientific data to demonstrate reasonable safety and performance. It is important to include appropriate treatment and control animals for evaluation. This may consist of positive, negative, sham, predicate, or reference device controls. Study time points should be scientifically based on known tissue responses or risks from the use of your device. Studies should be designed to include animal cohorts to evaluate tissue responses over the course of the study based on the expected duration of clinical use. For example, it might be necessary to evaluate both short-term and long-term responses for a permanent implant device. When selecting study size, it is also important to take into consideration the possibility of animal loss over the course of the study. Finally, an important aspect of the study design is to include adequate endpoints to evaluate safety and performance based on the potential risks of the device. For neurological devices, safety endpoints may include clinical observations, such as neurological and behavioral assessments, hematology and blood chemistry, and upon termination of the study, gross necropsy and histopathology evaluations should be conducted. It should be noted that there are various stains available for evaluation of tissue response or neurotoxicity. Also, as Dr. Ghosh mentioned earlier, certain elements of biocompatibility testing may be addressed in a well-designed animal safety and performance study. Performance endpoints may include device condition following use and device-specific functional endpoints. Additionally, labeling claims may need to be substantiated using the animal study. When evaluating your device, methods to minimize bias should be considered. For example, blinding of the study, the use of multiple evaluators, or randomization. It is important to include a thorough description of all procedures in your submission for our review, including administration of anesthesia and medication, clinical, macroscopic, or microscopic evaluations. This segment of the webinar is designed to provide a general overview of non-clinical evaluation of biocompatibility in animal studies in support of an IDE or pre-market submission. And we recommend that you contact our division using the pre-submission process to receive specific feedback regarding your device. FDA will review proposed biocompatibility evaluation strategies and animal study rationale, design, and protocols. However, please note that data cannot be reviewed through the pre-submission process. Additional information regarding the QSIP process will be provided later in this webinar. There are a number of resources available to provide information regarding non-clinical studies. As Molly mentioned earlier, the final biocompatibility guidance was published in June of 2016, and additional information regarding this guidance can be found in a related FDA webinar, which can be located through the CDRH Learn link provided at the end of the slide. The GLP requirements for non-clinical laboratory studies are outlined in 21 CFR Part 58, and there is a recognized consensus standard website that provides a searchable database for standards recognized by the FDA and the extent of recognition. Finally, a very useful resource is the FDA guidance document website, which provides a searchable database for both draft and final guidances, including some neurological device-specific guidances that may provide additional information regarding non-clinical testing and animal study recommendations for that specific device type. So that concludes this section of the webinar, and thank you very much. And our next set of presenters will be discussing the EFS program.
Thank you, Donna. Good morning, everyone. My name is Erin Keegan. I'm a biomedical engineer and lead reviewer in the division. I'm also one of the early feasibility study representatives for the division. So we're actually going to switch gears from non-clinical, and we're going to talk about clinical testing. And Devjani and I will be discussing our favorite kind of clinical testing, which are early feasibility studies, which is a kind of IDE study. So first I'm going to go through the definition of an early feasibility study and how it can potentially benefit you as a sponsor or investigator. Then I'll go through the stages of an EFS pathway to IDE and hit on some of the key submission milestones. Then I'll start going through some of the key EFS principles and some examples for our division. And at this point, I'll hand it over to Devjani to finish up key principles, go through some tips, and talk to you about initiating your EFS. So an EFS is a kind of investigational device exemption, as Carlos mentioned earlier. So it allows you to collect clinical data to support device safety and effectiveness. So an early feasibility study is a standard IDE, except it generally includes a small number of subjects. The device is generally early in development, so iterations are expected, and there may be limited non-clinical data, and enhanced clinical mitigations may be required. So it's important to note that EFS is an informal designation, and so you don't apply to be in the EFS program. If you have a very novel device and you want to get clinical data on a U.S. patient, then we will work with you as part of the early feasibility study program. So how can an early feasibility study benefit you? So it permits a more efficient pathway to U.S. commercialization because FDA feedback early on in your development may improve your development strategy and reduce unnecessary testing. Also, data collected in the U.S. patient population as opposed to maybe an OUS patient population may be easier to leverage to support later studies or marketing applications. The EFS study itself also enables collection of high-quality clinical data, and most of the time this will be the first time that the device is put into a human patient. And so with this data, you can optimize your device design or operator technique. You can refine the intended use population or non-clinical testing plan, and it can also help you develop subsequent clinical study protocols. So now I'm going to go through the stages of an EFS pathway to IDE. And these are all just recommended stages, but I'll start with, we recommend that you reach out to Devjani and I informally. This can be a phone call. And with here, we will talk about your device, We won't be reviewing anything technical. That comes later. But we'll go through the EFS program, help you kind of organize your initial submission. The next stage, again, this is listed as optional, but they really are all optional, is to do an informational meeting pre-submission, which is a type of pre-submission, and you can actually find more information about this in the QSUB guidance. But these are essentially just a low-pressure show-and-tell situation. You won't receive any feedback from the agency, but it allows you to familiarize the review division with your device and clinical indication. So then next you could submit your initial pre-submission, and this is actually outlined, the content, suggested content is outlined in the early feasibility study guidance. And then I really want to highlight stage 4 because the EFS process is highly interactive and collaborative, and so it's quite possible that you'll be submitting additional pre-submissions as you and the review team work through some of the non-clinical testing requirements and even the study design of the clinical protocol. And then finally, you submit your early feasibility study IDE, just like any traditional IDE, and we have 30 days to review your study. So now I'm going to go through some of the key principles of EFS as we've pulled from the guidance document, though I highly encourage you to actually read through the guidance document. You'll find a lot more information there. And I'll go through some examples that may be relevant to our device space. So the first principle is called just-in-time testing. And so this came about from the concern, or I guess motivation, that comprehensive testing on really early phase devices may add significant cost without return because you could be changing your device a month into your clinical trial based on feedback you've been receiving. And so we really want to stress doing the right testing at the right time. And this should not take the place of informative non-clinical testing. And these will be worked out in those additional pre-submission processes between the review division and the sponsor. So some examples could be deferring long-term durability testing, given the criticality of short-term benefit for devices such as glioblastoma, spinal cord injury, or severe psychiatric disorders, if may be appropriate. You could also limit the use of your device to a controlled setting, such as a hospital or clinic, instead of allowing the patient to take the device home in an early feasibility. This could change testing strategy for electromagnetic compatibility for certain electronic-based assistive devices. You could also use for a small number of devices, you may rely on single-lot ethylene oxide sterilization versus a full sterility validation. So this may be applicable for a novel spinal cord stimulation lead. The next principle I'll go through is the idea of using enhanced risk mitigation strategies in your clinical protocol. And so the concern here is that an EFS may carry greater unknown risks as compared to traditional feasibility and pivotal studies because these are just such new devices and we don't have as much information to evaluate the risk assessment up front. And so how we handle this is build in clinical monitoring right into the protocol. This could also include more frequent or detailed safety reporting to the FDA than you would see in a traditional IDE. And importantly, the informed consent should highlight the greater unforeseeable risk and the natures of these studies, because in the end, the patients need to understand that this is an early feasibility. So some examples could be if you have a stimulation device and you may be expecting some adverse events, the ability to titrate the therapy or turn the device off and revert to standard of care could be used. If you have a novel endovascular stroke intervention or just a novel surgical procedure, you can consider selecting expert surgical sites so that there's control in that aspect. Finally, if you have completed necessary biocompatibility and sterility, but you're still unsure about maybe the procedure or some aspect of the design and you want to monitor for infection, you could build that right into the protocol by having monitoring of the implantation site and the body temperature. So at this point, I'm going to turn it over to Devjani to finish up the EFS topic.
Good morning, everyone. As Erin had mentioned, I'm just going to finish up the remaining EFS slides. So one of the pillars of EFS is the ability to leverage data from non-traditional formats such as literature or testing from marketed products or earlier prototypes that you may have. It's important to understand that if you are considering leveraging data, you should provide a detailed discussion of the differences between the leveraged data and your proposed device. These are differences in the material, the device design, as well as the manufacturing process. You should also include a strong leveraging rationale, which discusses why, despite these differences, the data can still be used to support the safety and effectiveness of your device. Some examples of leveraged data that we have seen in our division is leveraging of biocompatibility endpoints from functional animal studies, as was discussed earlier in the biocompatibility section by Dr. Molly Koch. Data can also be leveraged from published literature. So, for example, with adequate justification, it may be feasible to leverage published spinal cord stimulation parameters to support stimulation parameters for a novel peripheral nerve stimulation. So another key EFS principle is timely device and clinical protocol changes. So devices under EFS may be early in development and is expected to change throughout the course of the study. This highlights the need for timely device and clinical protocol changes. Changes to the device or clinical protocol may be accomplished through two different approaches, the first being contingency approval. In this approach, basically device changes that you are anticipating during the study can be executed without necessarily additional FDA action if the proposed changes, the supporting test plans, and the acceptance criteria were agreed upon in the IDE or IDE supplement. There's also a broader implementation of the 5-day notice in the EFS IDEs. So traditionally, 5-day notices allowed changes in the device design that were not significant as long as it did not affect the safety of the subject. It also could not affect the validity of the data or the scientific soundness of the trials. These changes were allowed to be made prior to FDA approval, but the sponsor would need to inform the agency within 5 days of actually implementing the change. In EFS, the 5-day notice is a bit broader since EFS studies are associated with a small number of subjects and don't necessarily aim for statistical significance. So, for example, the sponsor may be able to change the effectiveness endpoint in the EFS study as long as these changes don't affect the safety of the study. We've also seen minor changes to device design, such as ergonomic modifications. In terms of contingency approval, we haven't seen many of these in our division, but some hypothetical scenarios may be the ability to adjust simulation parameters and possibly interchange prosthetic device components without FDA approval if the proposed changes have been agreed to during the review of the IDE or IDE supplement. So for the next several slides, I'll be presenting some tips that we often give to new EFS sponsors. So if you're making device modifications, we highly recommend that you record the device modification along with the rationale for why the modification was made. You should also record the testing that was completed with each device iteration. And you should keep samples of previous generations to help for leveraging in future submissions. So for example, you may be able to leverage some biocompatibility endpoints if your previous generation had similar materials and similar manufacturing processes. If you would like to use test results that were not obtained per standard FDA recommendation, we strongly suggest that you provide an explanation for why the data is sufficient. So for example, if your animal study deviates from GLP requirements, you should tabulate each part of the regulation, list how the study deviates, and how you will still be able to ensure the integrity of the data and minimize bias. The next tip deals with the use of short-term animal studies to support EFS initiation of a device that's intended for long-term use. So if you're planning to do this, we strongly recommend that you provide supporting evidence that short-term results are predictive of long-term safety. You should also consider applying additional clinical mitigation strategies such as longer-term follow-up. And you can also think about conducting additional non-clinical testing such as mechanical integrity testing under exaggerated use conditions, and perhaps also support your submission with computational modeling to support the long-term safety of your device. Please note that additional long-term animal data may be needed to support a larger clinical study. So when do you know that you're ready for an EFS? Well, it's generally when you can describe why additional non-clinical testing will not be informative and therefore a human clinical study is necessary. You can also justify how any leveraged information supports your clinical trial, and you can identify the potential risks and how they will be adequately mitigated. We recommend that you communicate these points to us using a device evaluation strategy as described in the EFS guidance, and the link is provided in this slide here. We're often asked by sponsors, when is a good time to talk to the FDA about EFS? Well, it's typically after you have established your general device design, you have an idea of your intended use, and what information you would like to gather from the EFS. And we strongly recommend that you come talk to us before conducting expensive and oftentimes time-consuming non-clinical testing. We also recommend that you reach out to the division EFS representatives, which is Erin and myself, to informally discuss the submission strategy. Our contact information can be found at the end of the presentation. Thank you for your time, and now I'll hand the mic over to Tim Marginin, who will present on FDA engagement and the pre-submission process.
Thank you, Dejani. So my name is Tim Marginin. I'm the Chief of the Neurostimulation Devices branch in the division, and I'm going to be covering a few key aspects of the Q-Sub guidance. The guidance covers multiple types of interactions. Today we'll focus primarily on pre-submissions, with also a little bit at the very end of my presentation about risk determination. So one of the big things that always comes up with regards to pre-submissions is the time frame for review. And per the guidance, we do try and hold a meeting if you request one within 75 to 90 days of when we acknowledge receipt. So we will provide feedback about 3 days in advance. If you request a meeting, you should generally plan to meet with us or receive that written feedback within about that time frame due to the workload considerations of review staff. So please budget your time accordingly as you're planning out how long you anticipate various steps in the process to take. Why engage as early as you can? Well, pre-submissions allow for potential issues to be identified earlier, and we can work through them with you as appropriate, particularly if you might have some type of novel technology or some sort of novel testing. You can always submit a supplement to get additional feedback if additional things come up, or if you revise something and you want our feedback on revisions. One of the common issues that we see with regards to pre-submissions, it's actually a fairly common issue across the spectrum of submission types, is e-copy. You really need to make sure that you comply with the e-copy guidance, because if you don't, your submission will not be officially logged in, the review clock doesn't start, and nothing else happens. I won't even know that it exists. And so there is an email address here that you can direct questions about the e-copy program to that email address. As far as submission contents are concerned, it's pretty basic and fairly general. Cover letter, background information, necessary information about device description, the types of protocols that you're running, whether it be venture animal clinical study protocols, if there are any, and specific questions related to that background information. One of the common issues that we see with regards to this is not enough information is provided up front. So a few years ago, we did an analysis of investigational device exemption letters, and we found that the area generating the most questions was actually device description, what the device is and does, instructions for use, the hazard analysis, and this is something that we see across the other submission types. If we don't have enough information to understand the device, we end up asking a lot of questions, and providing complete responses to those questions takes your time, and that extends the overall length of the review. So it's important to remember that you, as the applicant, know the most about your technology, not the FDA. The more that you can explain your thought processes when you submit the pre-submission up front, the more we can focus on the substance and give you better feedback. It's also helpful to understand the existing landscape as you're doing this. So please search for and review applicable guidance documents and standards if there are any, such as biocompatibility, which you heard about earlier, as well as other things like software, which applies to a lot of devices that come through our division. Make sure you explain the relationship of what you're proposing compared to what's been done in the past. As far as background information, it really is okay to err on the side of including more than what you think might be needed. Just make sure that it's organized and easy to follow. And if you intend to cite literature articles, please provide copies in the submission. There is such a thing as providing too much information. It's fairly rare. We don't need to see circuit diagrams for your devices. We don't need to see lines of software code. We don't need to see a copy of the entire grant that you may have submitted to NIH. It's good to avoid assumptions. Unless there's an applicable guidance document, an applicable standard, or other regulatory precedent that you can cite, it's a good idea to identify the most appropriate approach for your needs and to justify it. For example, not every animal study needs to use a non-human primate model. Some other type of model and protocol might be better suited to your particular situation. We also see some issues when it comes to specific questions. For example, not providing your own proposals for us to review. It's not really a great question to say, what animal should we use? Or how large should the sample size be if you're talking about a clinical protocol? We also get questions every now and then about wanting us to review data. Does FDA have any comments on the non-clinical test results? That's something that's left for the review of the actual marketing commission or an IDE, if that's the case. When it comes to specific questions, they should build on the background information that you've provided. So rather than saying what animal model should we use, you've obviously proposed one in the submission. What concerns do you have with our proposed animal model? It's helpful if you've said, here are some of the approaches that we considered. Here's why we think ours is the most appropriate for our situation. Another example of a good question, are the proposed sample size calculation method and related elements of the statistical analysis plan appropriate for the proposed clinical study? Again, getting us to comment on the approach that you have proposed. I'm going to finish up with a quick slide related to significant risk, non-significant risk, and basic physiological research with a focus on when an IDE is not required. An IDE is not required in several different circumstances. For example, if there are exempt studies for the IDE regulations, such as studies of approved devices that are used in accordance with their labeling, also certain diagnostic device studies. An IDE is also not required for basic physiological research, which is not for the purpose of evaluating safety or effectiveness of the device. We also don't need an IDE for practice of medicine when you're talking about the care of a specific patient with an approved device. And finally, we don't need an IDE for non-significant risk studies. The IRB can and should be making the first cut as to whether an IDE is required. However, if you still have questions about that, there is another type of Q-sub referred to as a risk determination request, which is outlined in the guidance document. You can send that in to us, and we will evaluate it, and the outcome will be a letter saying either that you're a significant risk, you need an IDE, non-significant risk, you don't need an IDE, or it's basic physiological research, in which case you would also not need an IDE. That concludes my section of the presentation. I am now going to turn the microphone back over to Dr. Pena, who will close out with some remarks.
Thanks, Tim. So a few closing thoughts. Our job at FDA is to ensure the safety, effectiveness, and security of a wide variety of medical products, including medical devices and neurological devices. Neurotechnologies represents an emerging technology area that we very much want to support and make our regulatory landscape as transparent as possible. Along these lines, we encourage industry, sponsors, developers, innovators to consult early with the agency, which offers FDA staff the opportunity to clarify the data needed to bring safe and effective products to market in the fastest way possible. And most importantly, the faster we are able to evaluate products that are safe and effective, potentially the faster patients and consumers can get the products they may desperately need. And I'd like to thank the staff in the planning, the execution, and the presentation of this webinar for FDA that is for this session. On the next few slides, we provide a primary point of contact for the center, namely DICE, Division of Industry and Consumer Education, as well as additional resources should you have questions. That is a very good place to start. And with that, I would like to close the presentation portion of this webinar. We are going to take a approximately 1-minute break, and then we will open the session to questions and answers. Thank you.
Momentarily, we will be beginning the formal question and answer session of the call. At that time, if you'd like to ask a question, please press star, then 1, and clearly record your first and last name. If you'd like to withdraw your question, you may press star, then 2. Again, to ask a question, please press star, then 1, unmute your line, and clearly record your first and last name. There will be a brief moment between each question. Thank you. We'll be beginning momentarily. We would now like to begin the question and answer session. Again, to ask a question, please press star, then 1, and clearly record your first and last name. Please try to limit it to 1 question per person. If you have additional questions, you may re-queue for additional questions. 1 moment for the first question, please.
me again. I might take the prerogative as this being an FDA webinar to ask a couple questions to our staff. And one question that we often get, that I've often received in conferences and in correspondence, is sort of like they never ask one question, they ask three questions. Is it ever too early to contact FDA? What is the best way to engage FDA? And who should I contact? And so I'd like for Tim to give a response, because I think those questions align well with the best practices to get us started.
Is it ever too early? Well, not really, unless you just have nothing done. So even if you're fairly early on in the process, and this is something that I didn't cover as part of my slides, but one of the aspects of the QSIP program is, and this was alluded to in the EFS discussion, was the idea of informational meetings. And those are a great way for sponsors to familiarize us, FDA, with your product, kind of where you are, where you're going, what you have, what you intend to do. Those are things that even though the timeline outlined in the guidance is 90 days, it's something where we can generally have a meeting a bit quicker, because there really is nothing for us to review at that point. It really is just kind of a show and tell. And if we have some off-the-cuff questions, we may raise those during the meeting. And those can be a good intro to FDA, and which can certainly be followed up with one or more pre-submissions for specific clinical and non-clinical protocols and things like that. The best way to engage is through things like that, taking advantage of the informational meetings, pre-subs, things along those lines, coming in, trying to come in just so you can understand as many of the expectations as you can before you get too far down the process and you run all these tests only to learn there at the end that, well, maybe that wasn't actually what we were looking for. The whole idea is to try and make the process as efficient as possible, and to make sure that everybody understands the expectations as you're going through the process. And the pre-submission process is a great way to do that. And then as far as who to talk to, well, if you happen to see somebody at a conference and you know that they're FDA, you can say hi. We're friendly people. If you have specific questions and we're not in a conference situation, but you have a contact here at FDA, let's say it's somebody in this division, you can send us a question. That individual may or may not be the right person to ask. If so, then they will try and answer your question to the best of their abilities as quickly as they are able to, considering their other work. Or if they're not the right person to talk to, they can certainly kick it over to who would be the right person. And if you don't really have a good point of contact, I will throw my name out there as one of the 3 branch chiefs. Branch chiefs are always a good point of contact to start with if you have a general sense for where your product would fall.
One additional question, I think. Studies is, one question that we do get is, I'm somewhat sure that I think I can go to an EFS study, but I'm not quite sure if I'm really ready to submit. How do I just talk through whether I have an EFS study or something other? And maybe Johnny and Erin, you can respond to that question.
So I think that's a pretty common question, and the best thing to do is just to reach out to Johnny and I. You can email us, and we typically will set up some phone calls. We do this frequently, actually, and we can discuss your device and what you plan, what you want out of the study. Actually, is an important question. So if you want a statistically significant large study, maybe EFS is not right for you. But yeah, it's pretty informal. We talk through some principles, and we help you plan your submission strategy. I don't know if Johnny has anything to add.
So one other thing is that for EFS, we typically, we try to see what the number of subjects are. So they're typically smaller studies. But there have definitely been instances where, you have a smaller study, but it may not necessarily be suitable for EFS because there's something very similar that you've already submitted as an IDE. So you don't need to go through the entire risk mitigation because that's already kind of with us and we have that on file, and it might be easier just for you to do a supplement. So there are certain nuances, and we highly recommend that you reach out to Erin and I to discuss this informally.
Without any further questions, I'd like to thank the presenters, the meeting organizers, and the many staff that were involved in the execution of this webinar. The information is posted online. Folks can take a look, and if they have questions following this session, be happy to address questions as needed. And we hope to share with you more about upcoming regulatory webinars in the neurological and physical medicine device space coming up next year in 2017, as it also relates in support of the White House Brain Initiative. And with that, I'm going to turn it back to Arlene.
Thank you. This is Irene here. We appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Thursday, September 22nd. If you have additional questions about today's webinar, please use the contact information provided at the end of the slide presentation. And as always, we do appreciate your feedback. Again, thank you for participating, and this concludes today's webinar.
This concludes today's call. You may disconnect at this time.